Hello all. My name is Eyyüb Selim Ünlü from Istanbul University, Turkey. Thank you for joining my session. My talk is on the study of Chikungunya virus protein sequence diversity dynamics. Let me provide a quick background. Chikungunya virus is an emerging arthropod-borne pathogen that causes a significant health threat. Since 2004, Chikungunya virus began an unprecedented global expansion resulting in major epidemics affecting millions of people. Different mechanisms are involved in the generation of viral diversity, which plays a prominent role in viral evolution, pathogenesis, immune escape, and drug resistance. This study focuses on protein-wide analysis of Chikungunya virus sequence diversity dynamics. Figure 1 shows the overview of the methodology workflow. In the first place, all reported Chikungunya virus protein sequences, a total of 5,820 sequences, isolated from humans, were collected from the NCBI virus database. Then, protein blast was done to group the sequences by the 9 proteins of the Chikungunya virus, which comprise of 4 non-structural and 5 structural proteins. Each protein dataset was manually checked and cleaned of any irrelevant sequences. Duplicate sequences were removed by use of the CD-HIT tool, and subsequently, multiple sequence alignment was carried out by use of muscle tool. In order to analyze the diversity in the context of the immune system, the bioinformatics analyses were performed on aligned monomer sequences. Shannon's entropy was calculated for each of the aligned overlapping monomer positions of the proteins. Furthermore, each position was analyzed for diversity motifs. Figure 2 illustrates the different diversity motifs. Here is an example of a monomer position with 20 sequences. The predominant monomer at this position, with a frequency of 12, was classified as the index sequence, with the 8 others considered as total variance to the index. The predominant variant of the index is termed as the major variant. Other variants with the multiple different sequences, each present in 2 or more of the aligned viral sequences, are called as minor variants. Each distinct variants at a monomer position are referred to as nonotypes to measure the number of different substitution variants. Figure 3 here provides the Chikungunya virus entropy plot and incidence of total variants. Entropy values are on the left y-axis, whereas total variants are on the right y-axis. Higher entropy values indicate higher diversity, and the lowest entropy value of 0 indicates complete conservation. Total variance is defined simply 100% minus the incidence or frequency of the most predominant sequence, which we refer to as the index. For example, if a position has a predominant sequence or index with an incidence of 80%, then the total variant incidence at that position would be 20%. The Chikungunya virus protein-wide mean entropy was as low as approximately 0.41, indicating that the emerging virus is highly conserved. Nonetheless, only approximately 70.4% monomers were completely conserved, with entropy value of 0. The most diverse protein was enveloped glycoprotein 3, exhibiting the average entropy of approximately 1.19, while the most conserved was the non-structural protein 2, which exhibited average entropy of approximately 0.22. The absolute peak entropy was observed as approximately 2.71 in capsid protein. For a benchmark, this line indicates the highest monomer entropy value that we have observed from our various analyses of viruses. The peak absolute entropy of 9.2 and total variance of 98% was observed for HIV-1 clade B. Figure 4 provides a scatter plot to better understand the relationship between the entropy and total variance. Each dot represents a monomer position. A positive correlation was observed with a wider spread in total variance with increasing entropy or diversity, particularly from entropy value of 1 onwards. So, an entropy value can have a range of total variance. For example, entropy of 1, represented by this red line, can have all these different total variant values. Given this, we then went on to investigate the diversity motifs that comprise the total variants, namely the major, minor, and unique variants mentioned earlier. Figure 5 provides a distribution for each of these motifs. The variant motifs displayed a characteristic pattern of incidence change in relation to increased total variance. In these plots, the x-axis is total variance and the y-axis is incidence. The incidence of the predominant sequence index ranged from 100%, which means completely conserved, to as low as 28.3%. Major variant, which is the second most predominant sequence at a given monomer position, showed a restrictive pyramidal pattern with a peak incidence of 49.1%, and thus closely challenging the index incidence. Notably, minor variants, the monomer sequence that were observed more than once, but of lesser frequency than the major variant, were on average below 5% incidence. While unique variants, which are the singleton monomer sequences or observed only once in the alignment, were present at most of the positions with non-zero total variance, with the maximum incidence of 19.6%, and thus these are unlikely to be a result of sequencing error. Nonatypes evaluate the multiple distinct variants at all the non-zero variant positions, with the maximum incidence of 25%. The violin plots here in Figure 5 show the distribution of the incidence values for each motif. In conclusion, this study provided important insights to Chikungunya virus proteome diversity, which can help better understand the evolution of the virus. Thank you.